Dear students, today we will discuss about the seed treatment, packaging and handling under seed technology course offered in fourth semester BSc degree programs. So today we will discuss all about the seed treatment, packaging and their handling. Seed treatment is very important activities and during in seed technology. It refers to the protection of seed from disease and pests. Actually, after processing the seeds, seed is treated with either fungicides or insecticides or in combination of both for the protection of seed against pathogens, insects and other different conditions. The objective of seed treatment is first to prevent the spread of plant disease. We know the plant diseases is spread by two ways, that systemic disease and non-systemic disease. We know very well that the systemic disease or those diseases which infect the plants during the flowering times, and in the non-systemic disease it is just later on it is infected the plants. So we just control this, we just protect for spreading of this disease by protecting them by using the seed treatment with fungicides. Second one is the prevention of seed from seed rot and seedling blight. Some of the disease which occurs right from the seedling stage or when we sow the seeds, at that time some disease may take place. So by protecting those seeds before sowing, it just protect the seeds from these two, reseed rot and seedling blight. Improvement in seed germination. Sometimes seed germination is improved by protecting the seeds with different means. Say, suppose the seeds are somehow not up to mark for this this purpose. So in that case, seed when treated with some rhizobium cultures or some other means, it is just improve the seed germination. Protection from storage insect. It is very much important, particularly for the seed treatment. In case of there are number of insect pests attacked to the storage seeds during storage, and so the storage conditions should be very much congenial to the storage purpose. Otherwise, if there is high moisture content and low humidity, and so this just favors the attack of insect pest in the storage purpose. So before storing the seeds, it must be treated with the fungicides, with the insecticides, etc. Last one is protection from soil insects. What we know that when seeds are sown in the soils, there are number of flora and fauna present in the soil which just try to infect or just damage the seeds in the soils. So in that way, the seed treatment is very much beneficial for this purpose. So the next slide is the types of seed treatments. There are different mode of action of the treating agent, that is maybe chemical, that may be other things for this purpose. So in that way, the mode of action seed treatment is of either seed disinfestation, seed disinfection and seed protections. Seed disinfestation is the protection action in which seeds are contaminated on the surface by spores of different pathogenic organisms, and in this case the organism is not infected the seeds inside the living cells. So destruction of surface borne organism is taken place in this mode of action of seed treatment. Next one is the seed disinfections. In that case, the pathogens are established themselves in the living cells of the seeds, and then we have to eliminate, hence we have to eliminate the pathogens which are penetrated into the living cells by different use of chemicals and other fungicides and other means. Fungicidal treatment must actually penetrate to this in the seeds. Here it is very much important that the mode of action of the fungicides must be such a way that it should penetrate into the seeds. And third one is the seed protections. It is nothing but only the covering of the seed with chemicals and other materials. So to protect the seeds and young seedlings against the pathogenic organism either under storage or in the soil is due to the seed protection. Condition. What are those conditions of seeds which must be treated? So there are different 4 types of seeds conditions in which it is essential to treat the seeds with chemicals and other materials. That if it is injured seeds, if it is diseased seeds, if it is undesirable soil condition, means if we have to plant, we have to sow the seeds in little bit waterlogging condition, little bit in acidic condition, or in saline condition, or in other conditions which are not favorable for proper germination. So in that case we must treat the seeds for proper up to optimum germinations. And last one is the disease-free seeds. In this case, in these all conditions where seed treatment is essentially required. Now come to the methods of seed treatment. There are different methods of seed treatment. It may either by mechanical means, it may be by chemical means, and by the physical methods of seed treatments. In case of mechanical methods, some of the seeds are altered in their shape and size due to the formation of the fungus sclerotia, presence of sclerotia or some other things. So in that case, these adulterated, or you can say that the altered seed type was just eliminated, just separated by the sieve or flotation of the seeds in water. It is just done. The best example for this control mechanical methods of this seed treatment is Agar disease and Tundu disease, which is controlled only by these methods. In chemical methods, we use organic or inorganic chemicals, maybe that mercurial or non-mercurials. In this case, non-mercurial, generally we use the organic non-mercurial chemicals like therum, captan and carboxyl. These treatments must be done at least 1 week before the sowings. We must note this, that the seed treatment by chemical means is done before sowing, 1 week of sowing, and also the seeds must be dried in dry places. Last method is the physical methods. In this methods, seeds are subjected to heat treatments. This heat treatment may be done either by hot water, either by the hot air or the solar energy treatments. So in this way we can treat the seeds under the hot water condition. In case of hot water treatments, particularly for loose smut of wheat and barley and oats, the seeds are first pre-soaked for 3-4 hours at 20 to 30 degrees centigrade temperatures, and then it is just exposed to 40 to 45 to 50 degrees centigrades of temperature for few minutes. So it controls the diseases of loose smut of wheat and barley and oats. In case of hot air treatment, it is meant for the controlling leaf roll virus. In case of potato, it is very much important diseases. It is controlled. It is controlled by the treatment of hot air treatments, in which a hot air boxes is prepared, and that in the potato is placed in that boxes, and the high humidity is maintained, and thus and the temperature is 35 to 40 degrees centigrades is given for the hot airs. So sometimes solar energy treatment is also done for the seed treatment purpose. In this case, seeds are just spread over the sun, in the sunlight, and dry places, and that the seed should exposed at least 40 degrees centigrades. When the temperature is 40 degrees centigrades, some of the diseases will be controlled by these methods. Seed treating chemicals, there are different protectants and chemicals which are used for this seed treatment, and first one is the seed protectants. These chemicals can be directly applied to the grains, and sometimes the formulation of chemicals having residual toxics or effects or repellent action on or both over the seeds. So in that case, applied directly to the grain to prevent damage by stored product, pest, for example clay minerals, red earth, kaolin before 1950. Now their use is restricted only to seed and grains meant for animal feed. These grains protectants can be applied in the form of a spray or dust on the uninfested grain. Characteristics: the protectants should have some good characteristics, that it must be effective for all diseases, it must be cheap and easy to handle, it should not be injurious to the human or the persons which are concerned with this seed treatments, and also not prolonged storage, not injurious for the prolonged storage. If the seeds are stored for longer periods, it should not be injurious for this. Stable in the package on the seed or in the soils. The package of the seed must be stable, and also the compatible with inoculants. When we store the seed in bulk, in that case there is very difficulties may taken place, that in that purpose a large quantity of seeds have to treated. So in that case, gaseous treatment is recommended. Gaseous treatments means fumigation. Fumigation is the treatment given in the form of gas under airtight chamber. They have the advantage of the rapid action against insect. The fumigation is very much effective when the airtight chamber is provided, and it should have high penetrability. The commonly used fumigants are solid form, that is sulfurs or quick phos we use generally for in the storage purpose, for in the fumigation purpose, and sometimes liquid form of ethylene and dibromide is also used for fumigation purpose when seeds are bulked stored in bulk. Effects of fumigation depends, so there are certain conditions which may affect the efficiency of fumigation, that the moisture content of the seeds. If there are high moisture content in the seeds, the fumigation may cause the injuries to the seed and the damage of the embryo. The concentration and dose of fumigants is also very much important, because if it is higher dose of fumigation is done in the storage, then sometimes it will be hazardous, it would be just dangerous for this seed. Duration of treatment must be done at standard, at optimum levels, optimum time, and dosage is also important for optimum dosage should be there. Frequency of the treatment, if it is required, it is needed that the once fumigation is done, but it is still needed to fumigate this stored because of this air passing or some other causes, then in that case it should also be repeated. And type of fumigation is a spot fumigation, which is generally done in the godowns and fumigatorium. These are two types of fumigations which are taken place during the seed treatment. The advantage, there is some advantage of seed protectants over the fumigants, that in case it is used as a polyphylactic treatment, prophylactic treatments, then effective when the grains are stored in loose containers where fumigation is not possible. We know very well that the fumigation is only possible when the seeds are stored in airtight chambers. But if it is loose, then in that case the seed protectant can only be useful for the seed treatment purpose. It is less dangerous than the fumigants because because it is just protected. It is not any type of gas is not releasing from the protectants, so it is not injurious to the human health hazards. Don't affect germination adversely, and sometimes fumigation of the seeds, if it is high doses, then in that case the embryo is affected, so the germination will be affected. So the next one application at harvest time is sufficient for 1 year. In case of seed protectants, if once we treat the seeds with the chemicals or by other materials, it is sufficient for protection of the seeds throughout the years. But in case of fumigation, it is difficult because of the leakage of gas from the airtight chambers by any means. Halogenation seed treatments, in this case protective treatments given to the seeds in the production cycle of most of the seeds. In this treatment, chlorine or iodine based halogen mixtures are prepared and treated with seed either dry or Slurry treatments. The preparation of chlorine based halogen mixture can be done with this procedures, that calcium carbonate is first dried in an oven at 100 degrees centigrade for 24 hours. Then bleaching powder and arapu leaf powder are mixed with the Ca calcium carbonate in the ratio of 5:4:1, and so these are some of the methods, some of the process which for the preparation of this halogenation seed treatment. Advantage of this halogenation treatment is that it gives protection against natural deterioration by reducing the free radical formation. Less harmful, it is less harmful to human and animal. It maintain high germination of the seed during the storage. Growth and vigor improved significantly. Protect the seeds from storage pests. These are some of the advantages of this halogenation treatment of the seed treatments. Overall, we can have the advantage, we can know the advantage of seed treatments, that it first protect the seeds from the insect pest diseases like seed rot and seedling bite. Like this, it improves the germinations, provides protection from storage pest. 20% loss is meant due to the storage pest. So by protecting the seeds with chemicals and some other insecticides or some other materials, we just save the seeds from storage pest. Controlling soil insects, the soils, nematodes, maggots, roots, grub, etc. will be not able to affect the seeds if it is treated with the chemicals or some other things. Addition, nutrition facility. See, these are some of the advantages of seed treatments. Some of the disadvantages side by side. It is some of the disadvantages of seed treatment is that there may be chances of accidental exposure of the worker who are producing or applying the seed treatments. Who are applying the seed treatments, it is accidental. Next, the contamination of food supply by accidental mixing of treated seeds with food or feed grain is possible. Accidental contamination of the environment also taken place through improper handling of treated seeds of or seed treatment can treated chemicals. All these risks can be can be avoided by taking precautions for during seed treatments. Next one is the equipments. What are those equipments which are generally used in the seed treatments? There are different equipments like slurry treaters, direct treaters, drum mixer, grain agar and soil. These are some of the equipments which are generally used for the seed treatment purpose. We will deal one by one this. In case of slurry treaters, it is a slurry is prepared by mixing the chemicals with water in a exact proportions, and the treatment material to be applied as a slurry is accurately measured through a simple mechanism comprised of slurry cup and that seed dump pans. The cup introduces a given amount of slurry with each dump of seed into a mixing chamber where the seeds are mixed thoroughly. So in this way we can treat the seeds with the slurry treated methods. Direct treaters are one of the equipment. There are two types of direct treaters, the panogen and mist-o-matic treaters. In this mist-o-matic treaters are generally very much useful, and it is generally used by the seed companies or some other agencies. So in this case, mist is directly sprayed over the seeds. The treater is equipped with large treatment tank, a pump and a return that maintains the level of a small reservoir from which the seed is fed. So this way we just use the direct treaters. Next one is the drum mixer. It is very commonly used by the individual farmers or the small or big farmers, this drum mixer. In this, mixer can be made a running pipe through a drum at an angles. The drum is mounted on the stand, and then in this the seeds and treatment are placed in the drum and it is rotated slowly under all the seeds are connected with the chemicals. The dose of chemicals must be optimum, which are recommended dose should be given. Recommended dose of chemicals should be given in the drum with the seeds and properly rotated slowly until the seeds are properly mixed. Then in this, the next one is the grain agar. In this, liquid material are dripped on the seeds as they enter a grain agar or straw conveyors. By the time seeds have left the agar, the chemical is sprayed on the seeds. The next one is the soil. Soil is the equipments which are generally used when the seeds are sprayed, when seeds are dried in the dry place, and in the thickness of this the seeds, the spreaded seed was 10 to 15 centimeter in depth. The proper amount of chemicals is diluted with the water and sprinkled over the seeds. The treatment, the proportion, the mixture, the water, whatever suspension is created, it is sprinkled over the seeds. Mixing is done with the soil, or the soil is one of the equipments which are generally used for turning the seeds all around by for 20 times. It is very much essential for proper mixing of the chemicals with the seeds. Some of the precautions must be taken in the during seed treatment, that most products used in the seed treatments are harmful to the human as well as for the seeds sometimes. So in that case, ensure that treated seed is never used as human or animal foods. Always must be, and for that indication should be there at the level that these seeds are treated with chemicals or some other materials which are injurious to the human health as well as the animal health. So it must be well labeled Treated seeds should be clearly labeled, like whatever treat seed are correct dosage rate and proper moisture content. Always we have to take care that the dose of chemicals and the seed must be optimum. Like if we are using the 2 gram per kg of seeds, that it must be stick for this. If the seeds are to be treated with bacterial culture, then in that case must be done in order that the chemicals must be first in the seeds must be treated with chemicals, then fungicides, then insecticides, and then this special treatment of the rhizobium culture or some other microorganisms is treated the seeds. So some of the other invariabilities of seed treatments are also done, and these are some of the special seed treatments are seed fortification. In this case, seed is treated with the minerals, vitamins and some other chemicals for proper vigor, for expressing the proper vigor of the seeds. So tomato seeds are soaked in vitamin E, it dissolve in acetone during the storage and have a good vigors. Seed infusion. Seeds that are susceptible to soaking injury and seeds with internal seed-borne disease which need penetration of chemical to kill the pathogen are soaked in alcohol, dissolve nutrients like vitamin, pesticides and can be dried quickly. So infusion of topsin theorem into vegetable seeds are generally used for this purpose. Seed hardening is also very much important. So in this methods, the germination seeds are improved, particularly in the arid regions where the seeds must be hardened. So it is a pre-sowing treatment of hardening of the seeds. It modifies the physiological and biochemical nature of the seed, protoplasmic characters, and increasing the physiological activity of the embryo and associated structure with the seeds. So this leads to increase in the elasticity of the cell wall and the development of a stronger and efficient root system in the seedling stage and later on of the plant growth. Seed treatment with biofertilizers like rhizobium, azosporium, must be done before sowing. But always remember that it must be in sequence, that FIR should be followed. That first we treat the seeds with fungicides, then insecticides, and then lastly the rhizobium. Pelleting of the crops, it is also very much important when the seeds are very small in size and shapes. In that case, the seeds are enclosed with the filler material which material using the adhesive just large enough to produce a globular unit of the standard size. Just increase the size of the seeds for proper planting. It is just seeds are pelleting with some filler materials to facilitate precision planting and to serve as the mechanism of applying needed materials on the seeds is done in this pelleting crops. And another one is the seed coating. These two are very much common, but difference that the seed coating is a technique in which additives such as pesticides, nutrients or nutrifying bacteria are applied to the seed coats. But in contrast to pelleting, the coating conforms to the individual seed shape and it does not significantly modify the seed size. The coating is done in such a way that the increment in the size of the seed is not done. Whereas in case of seed pelleting, the seed size is increased for the standard size of the planting. Next, seed coloring. Seeds are colored with the non-toxic dyes like methylated bromoresol green, methylene blue and rose Bengal dyes, it all are the chemicals which are generally used for the coloring purpose. Particularly the seed coloring is done particularly for the identification of the seed lot, identification of the particular variety, identification of the brand, and sometimes it is very much useful in the identification of A, B and R line. We know very well that A is male sterile line, B is maintainer line, and the R is the restorer line. If these three lines are colored with different color, so in that case we can identify easily that this is the sterile line and it must be nearby shown to the restorer lines. So the color faded seeds sometimes is also used. Color is also used for the faded color seeds to increase the figures of the seeds. Seed tabs, these are the tabs embedded in paper or gel. Pre-treated seeds are placed equidistantly in the gel and then cooled. So these tips can be rolled out in the field covered with the soil and water. So these are some of the advantages of this additional treatment, your special treatments, seed treatments, that uniformity in seed size like this. Next one is the seed packaging. After seed treatment, it is very much essential to pack the seeds properly. So the seed packaging is a process in which filling of the seeds with a specified quantity is done in a particular containers. So it is done just after seed processing or seed treatments. Then seed packaging is done. This consists of the following operations: that filling of seed bags to an exact number. It may be automatic, say manual, automatic or semi-automatic. So placing leaflets in seed bag regarding improved cultivar practices is essential. This is essential to have all information about during the packaging. The seed labels should also be attached properly. That must be attached and have the information about the seed lot, about the class of seeds, about the variety. All these information must be mentioned on the labels, and it must be attached on the seed bag with the CV. A storage of the seed bag is done properly. So after this, some of the equipments which are used during packaging is the baggerware, the bag sewing machines. Baggerware is the small machine which are mounted just beneath a bin, and then in that case, in the baggerware, the empty bag is attached to the machine by a big bag clamp, and the actual amount of seed, the specified quantity of seeds is flow in the bag, and then it is tripped, the levers. So the tripping the lever may be manuals, semi-automatic or automatics. So seed flow lever tripped automatically after a specified weight. Filled bag removed from the baggerwares, and then baggerwares and the bagging scale may be manual, semi. So this baggerware means when it is filled and then it's sewed, and then it is just weighed by the scales, different scales, mounted on the processing plants. So next one is the bag sieving machine. Is very much important, that top of the filled bag sewed by high speed precision sewing machines. The sieving of the bags must be done properly. Otherwise, if there is any leakage, then seed maybe just lose out. So in this case, it must be done properly. That sieving machine must be equipped with the thread properly, with proper size. Operation by the trained personnel is done. In case of plastic bags, if the bag is plastic, so in that case sieving is not done. So in that case, the heat sealer is generally used. And then the lastly the weight the seeds packed and sealed and packed as just weigh on the platform scale balance. Elevating and conveying equipments is used for moving the seeds from processing plants. It is convey the available seeds from in either in vertical or horizontal or some inclined directions. In this case, the selection of this conveyor is very much important as for the effective and efficient processing. The types of conveyor generally used in processing plants are bucket elevator, belt conveyor, vibrating conveyors, and chain conveyors. Some of the factors which must be considered while packaging is done, that the kind of seeds to be packaged, quantity of seeds, it should not be too large, value of the seeds, the seeds is, if the seeds are costly, it must be packaged properly with the cloth or plastic bags. Cost of packaging materials should also be considered, because if it is high materials is used for packaging purpose, then the cost of the price of the seed will be increased. So storage environment is also essential for considerations. Period of storage, how much time the seeds are being stored, so it should also be considered for during the packaging and transport of the seeds. If it is which means the seeds are being transported from one place to the other place, it is very much important. If we pack the seeds in plastic bags, then in that case there is chances of damage during transportation, so it must be taken care. So some of the recommended seed packaging bags of the foundation seeds is like, with the respect to the crops, we can see here that for the paddy seeds we use only the cloth bag, and having with the size of 58 to 40 into 43 centimeters for the net weight of 12 kg only. So here the specified quantity of the seeds is mentioned against the different crops and the type of bags. In case of maize, the polythene in the cloth is used with the size of 58 into 43 centimeters, and in that case 10 kg seeds are only packaged. So in case of gram, it may be packaged in cloth bag or in plastic bags with different size and different weight. The procedure of constructing seed lot, it is very much important, because until unless we don't have the seed lot number, we cannot trace back of the particular seeds. So in that case, lot number, designation of lot number is very much important. Seed lot number have generally 4 parts. First part is the month and year code. So it represents the harvesting time of seed crop, abbreviated like JN, January 2000. It means the seeds are harvested in the month of January in the year 2000. This is the first part, or you can say that month and year codes. Second part is the production location codes. Production location code means that the place, the location where the seed has been produced. So for this purpose, there are numerical given by the central government, that for example, for 1 is given for the Andhra Pradesh, 2 is given for the Arunachal Pradesh, 3 for Assam, 4 for Bihar, and so on, 32 for Pondicherry. So these numerals has been designated as the production location codes. So we can identify that this particular lot is produced in the particular area, particular state. Third part is the processing plant codes. Generally, in each state there is certifying agencies. The establishment of certifying agencies are there, and these certifying agencies just designate a numerals to the different processing plants available in the particular area or state. So it is again, it can indicate the seed processing plant where the relevant seed has been processed. We can trace back that the seeds where the seeds has been processed. And fourth part and the last part is the seed produce code. It is the ultimately serial numbers of the any individual lots. If the seed lot size is more, then it may partition in the different weight with different weight, and then we can use the Roman numerals for this. If we consider the example that the seed lot number, if seed lot number is May 88-12-01-01, it indicates that the first code as first part, that month and year codes. So the seeds are harvested in the month of May during the year 88. Second part is the production location code is 12. It means that this particular 12 is designated for the state Madhya Pradesh. So it can be traced back, it can be identify the particular seeds is produced in Madhya Pradesh state, and 01 is the processing plants. The processing plants, the seeds are processed or in the state of Madhya Pradesh, which have been designated as 01, and the last one is the ultimate number of the produce. So this way we can have the lot. Now we can prepare the seed lot number for tracing back the seed. Is all about the seed treatment, packaging and proper handling. Thank you very much.